hydrophobic hormones. So we talked about hydrophilic hormones. They're able to dissolve easily in water. The extracellular environment is mostly water. Therefore, you find their receptors on the outside. But what about hydrophobic hormones? Well, hydrophobic hormones, again, lipids are hydrophobic, and cholesterol and steroid hormones are lipids. Therefore, they are more likely, they aren't going to behave like your hydrophilic monoamines and peptide hormones. They behave in their own special way. So most of your plasma is water. So if lipids are hydrophobic and cholesterol and steroid hormones are lipids, what prevents them from just separating it like we see in this image right here? So how do you dissolve steroid hormones in blood? Now, if you're taking biochemistry, things like lipids, they can dissolve in water, but very, very, very tiny amounts. You can't have a whole bunch of like hydrophobic compounds dissolving easily in water. But so how do you transport them if they have a hard time dissolving in water? What you do is like when you have oil and water suspension, how can you get them evenly suspended? Well, you can make a mixture. Or if you've ever seen like the salad dressing in the bottles and or seen a really good salad dressing, it doesn't typically separate. Yep, so things like, yes, someone said lipoproteins. So you have a colloid is a solution of very large organic molecules. So you have something called emulsifiers, and I italicize this because that's kind of like almost like borderline chemistry, biochemistry. But these stabilize hydrophobic compounds in water-based solutions. So lipoproteins are an example of that. So what they do is that they're able to, like serum proteins, lipoproteins, bile acids. So this is how you're able to your blood is able to transport hydrophobic compounds. What is the cool thing is that these things like lipoproteins or bile acids or other types of proteins we call globulins, they're able to, overall, they're hydrophilic. They're able to dissolve in water. But they have certain hydrophobic sites or parts of them that are hydrophobic. So basically, you can have a hydrophobic compound or hormone binding to that one hydrophobic region of a mostly hydrophilic protein, and therefore it can kind of piggyback and be transported throughout your blood. All right, so then we're talking about hydrophobic compounds. Now, remember, so in the previous slides, I said that there's also intracellular hormone signaling. So things that lipid-soluble hormones, they freely diffuse into the cell. So compared to like a hydrophilic hormone, that actually has a harder time entering a cell. But when you have something that's hydrophobic and lipid-soluble, it actually has an easier time passing the plasma membrane and getting into the cell. So why can they diffuse freely? Well, this is where you have to go back to chapter 3. I think the cell biology chapter. So hydrophobicity, so if you remember the plasma membrane, it's not complete uniformly hydrophilic or hydrophobic. There are hydrophobic regions, there are hydrophilic regions. So why? Because these phospholipids, they're actually very interesting. They have these hydrophilic heads that like to point toward water, and you have the hydrophobic regions right here. Yeah, so this part, so the interesting about the plasma membrane, it's kind of like a hydrophilic hydrophobic sandwich. So you have your hydrophilic parts facing the water base outside of a cell and the water base inside of a cell. But in the middle, you have this hydrophobic core, and this is a big barrier that prevents a lot of compounds from diffusing from the outside toward the inside and vice versa. Now, the phospholipid bilayer, so you have the water base outside and you have the water base inside. This hydrophobic region and core, well, the interesting thing is that hydrophobic compounds are allowed to because they can dissolve into this area right here, this is why they're able to freely pass from the outside toward the inside and inside toward the outside. So hydrophobic compounds, they have an easier time passing through this hydrophobic core. So again, if this is all news to you, go back to my previous lectures in cell biology. So what can diffuse through the lipid bilayer if it's very small versus something that's very large? Nonpolar molecules don't have strong charges, and highly polar chemicals. If you don't know what a polar bond is, chapter 2 and chemistry. And then most sterols and steroids, so they tend to be they tend not to have a strong electrostatic charge. And then we have ions that cannot diffuse. So what I'm getting at here, so Sterols and steroids, because they don't have strong charges and because they're mostly hydrophobic, they're able to diffuse across that plasma membrane surrounding your cells. Now, steroid hormones, again, they're derived from cholesterol. This is why on the whole, steroid hormones are mostly hydrophobic. And cholesterol is a lipid. Lipids are hydrophobic in general. So when you have something that has a lot of charge, like an ion, these have a hard time passing through the hydrophobic layer. Something that's big and charged, like a glucose molecule, that's going to have a hard time passing through the hydrophobic layer as well. But when you have something that's hydrophobic itself, like dissolves like. So here we have the hydrophobic layer, here we have a hydrophobic steroid. So the steroid pretty much has free pass through the hydrophobic layer. This is why steroid hormones are able to cross through the plasma membrane and also cross past the nuclear envelope and the membranes that surround the nucleus into the nucleus itself. Now this is the martini version of so what we have here on the left is a steroid hormone, and here we have a thyroid hormone. Again, this is why I always put that little asterisk, and you should always highlight this part as well. Thyroid hormones are originally made from amino acids, but what do they behave like in the end? They actually behave more like a steroid, even though they're not made from cholesterol. When you have a thyroid hormone that's fully made and synthesized, it actually acts a lot more like a steroid hormone. Now, there's a lot going on here. Well, what do we notice here? We have hormones, and like I just established in the past few slides, hydrophobic hormones have an easy time passing from the outside of a cell, diffusing across the plasma membrane and going to the inside of the cell. So what we see here are that steroid hormones, you can have receptors in the cytoplasm. You can also have receptors that are already located inside the nucleus itself. Thyroid hormones, this is kind of like misleading. So you can have either receptors on the mitochondria, or you can have receptors that are already located on DNA. Now here's an example of a steroid hormone. So here we have estrogen receptor alpha. So here's a receptor, and it's located in the cytoplasm. And here we have estradiol, an example of an estrogen. So what we have here is that, remember that steroids, they can freely cross the plasma membrane because they're hydrophobic. So what happens is that estrogen crosses the plasma membrane, binds to its receptor, estrogen receptor alpha, in the cytoplasm. So estrogen receptor alpha actually likes to pair up. Like once it becomes activated by binding to estrogen, it likes to pair up with another estrogen receptor that bound to estrogens, and then it moves into the nucleus together. Then it binds to, now you don't have to know all of this specific stuff. This is actually from I believe my master's thesis defense. But what happens here is that it binds to DNA. So if you don't know what DNA is, again, go back to the cell biology chapter or my lectures. And what happens is that this activates these genes that become activated in response to estrogen receptors. So what happens is that basically they cause these genes to become active, and these genes make a protein in response to estrogen receptor alpha, or estrogen receptor activity, I should say. Now, thyroid hormone is very interesting. So the receptors, instead of being floating around the cytoplasm, thyroid hormone receptors or the non-mitochondrial ones, they are located in the nucleus, and they're already bound to DNA. So the interesting thing is that here we have a thyroid hormone, and it's going to bind to thyroid hormone receptor. Don't worry about what RXR is. That's more molecular biology stuff that I don't think you even cover that in either of the books, so don't worry about that. Here's the thyroid hormone receptor. It's already located on DNA. And what does this do? This activates genes. That's all I really want you to know at this point. So here's the easier version of things. So what we covered is what did we see in common between the two steroids and the thyroid hormone receptors and their signaling. So basically, lipid-soluble, these hydrophobic hormones, they're able to cross the plasma membrane. This applies to both or actually it's interesting because like thyroid hormone actually has a transporter, but it's still lipid-soluble. So what happens here is that, okay, lipid-soluble hormones, they can cross the plasma membrane, and then you have these receptors, instead of being on the outside of a cell, because these hormones are able to travel across the membranes, These receptors are located on the inside of a cell. Now, what happened in the end? They both bound to DNA. So when they bound to DNA, they activate genes. These genes, there are stretches of DNA that basically contain the instructions on how to make a protein.